So the paper I'm going to be presenting is Microneedle Array Delivered Recombinant Coronavirus Vaccines, Immunogenicity and Rapid Translational Development. So essentially what this paper is, it's about the coronavirus. What's not known is that there's actually several types of coronaviruses. You have the SARS-CoV, and this hasn't been around since 2003. You have the MERS-CoV, which is a Middle East respiratory syndrome, and it's found in 2012. And then that has actually a 34.4% fatality rate. And then you have SARS-CoV-2, and this was first reported December 31st, 2019. It stands for Severe Acute Respiratory Syndrome Coronavirus 2. This is essentially COVID-19. The difference between this name and COVID-19 is that COVID-19 is the disease and SARS-CoV-2 is the name of the virus. These researchers look a lot into MERS-CoV because that's what they were studying before SARS-CoV-2 actually came out. As you can see, this little diagram to the right kind of gives you an idea. Obviously, it's not fully up to date, but it kind of shows the differences between each of the coronaviruses. But you'll have to keep in mind that MERS-CoV and SARS-CoV are slightly different. And like I said, some of this research is based off of MERS-CoV, and then they switched to SARS-CoV-2 when SARS-CoV-2 kind of came about. So how coronavirus actually infects. Speaking of SARS-CoV-2, coronavirus has a spike protein, and that is actually this thing on the right here in this little cell. And it looks like a little golf tee is how I think it looks. And while it's on the cell, it exists in a trimeric form. And then within a eukaryotic ectoderm, or even when the S1 is active, it exists in a monomeric form. The S protein has two subunits. It has an S1, which is essentially just a receptor binding domain in the N-terminal surface, and then an S2, which is a C-terminal transmembrane subunit that fuses with host cell membrane. From the previous work with the MERS coronavirus, it shows that using the S1 subunit produces more effective results than binding the entire whole S protein to create a vaccine. So that's actually kind of what they shift towards the end here when they transition to SARS, is they simply just use the S1, and that's actually what most of the study is over the MERS-2, is just using S1. Also, it's important to note that the SARS-CoV-2 binds to the ACE2 receptors in our bodies. So some terminology here. mNA, that is a delivery method. It's microneedle arrays, and basically it's used for vaccinations. It delivers high concentrations of the vaccine in local skin microenvironments. So essentially, it's just like a lotion or a gel that you put on your skin, and it gives us the vaccine similar to that of like the normal injection way now that you would get a flu shot. They found that it actually works a lot better because the concentration is so much higher when you inject it onto your skin like that. There's the PAD, that is just a shuttle vector. Then you have the AD5, and this is a replication defective adenovirus. It's used for some positive control, and it's seen kind of later in some slides here. I'll go over it a little more. Then you have the S1F, the S1FRSO9, S1FFIC, S1F plus MPLA. So these are going to be seen at the end of the SARS or the MERS, and essentially it's different types of things that were ligated to help with stability and things like that. S1F is a fusion to a T4 fibritin fold-on domain, which helps keep the trimeric form. S1FRSO9 is integration. It integrates a TLR4 agonist peptide, and this helps with kind of some immunogenicity. S1FFIC is integration of Salmonella typhoon flagellum C, and again, it's just kind of hopefully to help with binding of antibodies and things like that. And then you have the S1F plus MPLA, and that's just sterile monophospholipid A, and essentially what that is just a normal injection method that will be used, and they test out that way as well in some of the MERS studies. So some methods here, construction of the adenyl vectors and plasmids. Basically, they attached a His-60 onto the virus, and they kind of constructed it and made it their own way. I will show in the next slide the actual constructions because I can't really explain how they did it. Purification of recombinant proteins. They basically used the His6 tag to purify through affinity chromatography, and they treated it with ACTEV protease, and that removes the histidine tag. The SDS page in the Western blot, essentially that's just going to be your normal Western blot, which we'll take a look at one of those on the next couple slides. Fabrication of dissolvable microneedle arrays. That is essentially putting the SARS-CoV-2 S1 vaccine or the MERS attached to the mRNA, essentially. So the mRNA is actually like a hydrogel, and I don't know if many of you remember my presentation earlier, but the hydrogel, it's a delivery method that I talked a lot about in my last presentation, so it's kind of cool that they're using that. The animals that they use for the SARS, it's 5 female mice inoculated with mRNAs loaded with SARS-CoV-2 S1 or SARS-CoV-2 S1 FRS09. They use some different type of mice for the MERS, but we're not going to touch too big on that. Then you have the FACS analysis, which is fluorescence activated cell sorter that screens for the MERS S specific antibodies in pooled sera. And then there's an ELISA assays. There's quite a few ELISA assays, which is collections of the sera that were obtained every 2 weeks and tested for MERS S1 protein specific IgG. And then finally, there is a neutralization assay, and that's just for the MERS-CoV. And that is basically sera obtained from retroorbital plexus every 2 weeks, 2 and 6 weeks, and was tested for their ability to neutralize the MERS-CoV. They didn't actually get one of those done because it needs to be a lot longer, a lot more reliable with the SARS, but they plan on doing that in the future. Again, I'll talk about it later. And then there's just some statistical analysis they use, which involves computers. So this slide is the construction and characteristics of recombinant proteins. This is basically for MERS specifically. This is kind of the first half of their research. As you can see here in figure A on the left, you have the MERS constructions that they made. You got that MERS S1 down here at the bottom that's secreted as a monomer. Then all the other ones, the MERS S, S1F, S1RS09, S1FFIC, those are all secreted as trimers. And you can see at the top here, kind of towards the top, is the actual MERS S, which is a membrane-bound trimer. So that's kind of a cool way that they demonstrated that there. And then over here to the right, you have figure B, which is a Western immunoblot. And you can see as where the monomers and the dimers and the trimers kind of ran because of that blot. So figure B, the lanes 1 and 5 are just mock lanes. Lanes 2 and 6 are 85 MERS and then SIF. Lanes 3 and 7 are 85 MERS S1 FRS09, 85, and then lanes 4 and 8 are the 85 MERS S1 FFIC. And then down here in figure C, it just essentially shows, I'm not going to talk too much about it, that the 3 MERS 1 proteins had strong reactivity to a MERS S1 antibody, and that's basically what is seen here in the binding down here. So here on the next slide is the detection of membrane-bound MERS S specific antibody. Figure A, I decided to include it on there in case you were curious as far as what their timeline was. It's pretty straightforward as to what that is. You can check that out in the paper if you want to. Figure B is just a flow cytometry just to ensure that all the cells basically had the MERS constructs that they had made, and you can see that they all in fact do have them, and they use a PAD vector as a control. We can go ahead and move to the next slide there. So induction of humoral immune response to MERS S1. So this is actually the bread and butter of the MERS S1. It's really cool. So using the ELISA assays here in this A and B at the very top, you can see that the MERS S1F and MERS, all these MERS constructs, after initial vaccination, then 2, 4, and 6 weeks here, you are getting IgG you're getting IgG activity for MERS S1 specific antibody. And as you can see here, and that was done through injection. And then over here, this is using the mNA. And basically here with the mNA, you're getting even higher antibody being produced, which is kind of indicating how well the mNA actually works. Figure C and D, it's a little confusing, but essentially what it is a neutralization assay, a MERS-CoV. And basically, it's what it here, as you can see, its ability to neutralize the CoV. And again, you can see with the mNA, it has a much higher ability to neutralize the CoV compared to that of just natural injection. Again, it's kind of proving that mNA is a great way to inject this vaccine into people and hopefully get a good immune response. So this slide is actually probably one of the most interesting parts of the paper, the longevity of the immune response. They tested it at week 6, 23, and 55. And in just natural type of injections, you can see in figure A here that when injected like that, you're getting an immune response basically all the way up to 55 weeks, but it did kind of decrease here at week 23. They didn't really expand on why that was, but the big thing here is when you have that mNA as your vaccines, you get a constant and even rise of IgG specific antibody for MERS all the way up to 55 weeks. So it's showing that this vaccine is actually being super effective and that antibodies are being created for this MERS. Okay, and finally, the SARS-CoV-2 testing. Again, this is probably what everybody was waiting to hear. SARS-CoV-2, they basically constructed they had the S protein here in figure A that you can see. It has a ribosome binding domain right there. It's got the S1 and the S2 subunits. And when it's membrane bound, it's a trimer. And they took a SARS-CoV-2 S1, which was just going to be the secreted monomer. And then they also constructed a SARS-CoV-2 S1 FRSO9 to have a trimer form of that as well. So that's essentially what the construction of the gene looks like. And then down here in figure B, you have another Western blot that essentially shows you're in lanes 1 and 3, that's your SARS-CoV-2 S1, and then lanes 2 and 4, that's your SARS-CoV-2 S1 or SO9, and that's basically showing the trimers versus the monomers there to prove that they are there and that they're super stable. They also noted in their paper how stable they were just because they would heated them and boiled them and put them through a lot of tests, and they actually never lost their shape. Especially the trimers, obviously monomers, but the trimers especially. And then here in figure C, you can show that the testing that they've done so far, and they've been able to test up to 4 weeks. So again, they don't have a neutralization assay to test done yet. But here, the control, it shows no IgG production in an ELISA assay. And then here with the mNA SARS-CoV-2 S1, so basically just the monomer form, you are getting this immune response up to 4 weeks. As you can see, at week 2, they do a boost. So basically, it's just a booster type of injection. So they give it again, and you get this IgG response here. And then on the far right, you get the mNA SARS-CoV-2 with the S1 RSO9, and you get the same type of response. It actually continuously grows the IgG. I would have to say the reason that they are trying the trimer and the monomer form, they didn't really specify in the paper, but what I kind of interpreted from what they had said was that they're trying it this way because of we know that it secretes as a monomer form, but our body could possibly bind the trimer form. So essentially they're trying a monomer and a trimer form just to see which one is going to work better to create a vaccine. But that is it for the SARS-CoV-2 testing that they have done so far. And the paper, you can go look, and they have kind of a layout of what they're going to do next and everything, and I'll kind of discuss that here in a minute. So the conclusions and some future work. Its successful development of an mRNA SARS-CoV-2 vaccines capable of generating a potent antigen-specific antibody response. So they have successfully done this and seen that the antibody response, because of their ELISA assays, again, it's only up to 4 weeks, and that's why they continuously say in the news that this could take up to years, up to a couple years or a year to have an actual vaccine out. And then another thing that they found out was the mRNA provides a very low cost, easy, and effective way to get a vaccine to the public. My kind of interpretation on this, and I honestly think it's so cool because imagine if mRNA was actually how they give the vaccine out. I mean, essentially, they could have it shipped to people's houses, and if you just rub it on your body, and now you're vaccinated. Obviously, it would have to be with a kit and everything, but it could be a very low cost, super easy, effective way to get the vaccine out to the public, which is desperately needed right now. And then they still need to, for some future work, they still need to perform more ELISA assays and neutralizations assays um, to validate basically further that this does in fact work. And then the last slide is just my conclusion. So thank you.